Hello, my name is Heather Jackson and I am very proud to present my presentation on extrinsic fluorescence. This is for class Chem 580 at Indiana University and it is our final project. So, what is fluorescence? That is such a great question. Typically, we think of fluorescence as something that glows. So, how is that useful in chemistry or biology? Well, fluorescence is combined with spectroscopy, and that combination of those things can give amazing results. Fluorescence is a type Fluorescence spectroscopy is a type of electromagnetic spectroscopy that analyzes the actual fluorescence or the glowing from a sample. This type of spectroscopy involves using a beam of light, usually ultraviolet light, that is very powerful. When the light is aimed at the sample, it excites the electrons in the molecules of certain compounds, and that excitation causes them to emit light as energy, typically, but not always, visible light. A complementary technique to the fluorescence spectroscopy is the absorption spectroscopy. In the special case of a single molecule fluorescence spectroscopy, the intensity fluctuations from the emitted light are measured from either single fluorophores or a pair of fluorophores. So, how does that work, you ask? Well, great question. Fluorescence is governed by three important events, all of which occur on timescales separated by several orders of magnitude. Although these timescales are separated by several orders of magnitude, they are very, very small, micro and nano portions of a second. So, first, the susceptible molecule is excited by an incoming photon. And you can see that right here in these green arrows that are showing here. It's going from the ground state to the excited state right here. The absorption is 10 to the negative 15 seconds. Next is the vibrational relaxation of the excited state electrons to the lowest energy level. Right here are the yellow arrows here are the internal conversion and vibrational relaxation. Lastly, the emission of the longer wavelength photon and return of the molecule to the ground state here. And as you can see, that is where the fluorescence is happening. It makes it move to a excited triplet state, and this is where you can see the fluorescence happening. Fluorescence is a stunning manifestation of the interaction between light and matter. It forms the basis for several different things, but mainly for steady state fluorescence spectroscopy, which is where you can look at things in a steady state. Time resolved fluorescence spectroscopy, which means you can see how things are moving and what they look like, and microscopy. The photo at the top is an example of a portion of a molecule that is undergoing fluorescence. These types of fluorescence are tremendously sensitive in emission profiles. They give high spatial resolution and high specificity of fluorescence. This amazing specificity is causing the technique to quickly become an important tool in genetics and cell biology. There are advantages and disadvantages of fluorescence spectroscopy. Advantages. First, it is very sensitive, so you can easily see different types of fluorescence. Secondly, it can be used for quantification of fluorescent species. You can actually count the different things. Third, it is easy and quick to perform the analysis. It does not interfere with normal biological systems, nor does it destroy the sample. What are some of the disadvantages? Well, it's not very useful for actual identification. Secondly, not all compounds fluoresce. Sometimes you can put the light to the compound with a probe attached to it and there is no fluorescence. And thirdly, contamination is an issue and it can quench fluorescence and give false or no results. And when I say quench fluorescence, it means that it prevents it from being able to fluoresce, and then you get a negative or false result. The amazing thing about fluorescence is that it is actually a combination of the chemical and optical recognition, and you can use your eyes to look and see what is happening on the chemical level. An analyte is first formed. It's an isolated or complex dynamic mixture. In the example in this diagram, it looks like a double helix. It is combined with a probe, which is a targeting group that has organic dye. And then that dye is actually fluoresced. And when it is fluoresced, that's when you get the optical connection. Your eye can see those colors, or the spectroscopy can see those colors, and it takes it to a close-up of the retina tissue on the right-hand side, which shows what is happening as those different colors enter the eye. You can see them. Analytes can be evaluated by a wide range of environments. On the left-hand side here, you can see that we have a blue fluorescence, a green fluorescence, and a red fluorescence. And when we put those all together, you can tell that the blue is actually chromosomes, and the green here are the little threads that are hold those chromosomes together during meiosis, and the red is the outside part of the cell. When you put it all together, you can get a picture here of what the chromosomes and the spindle fibers in the cells look like during mitosis. This picture here is a photo of DNA. Obviously, it is in the form of a double helix, and it is being fluoresced a blue color. And then there's different pieces of cells that are being fluoresced using different colors so that you can see the cell anatomy and what is happening in those cells at that point in time. All right, so here's an example of how fluorescence can be used. This photo is 1 micron spheres, and this is being used under transmitted or incandescent white light, what you would normally see in a microscope if you were just, shining the light through and looking at it. The light passing through the sample is diffracted, partially absorbed, and transmitted to the detector. All right, if the detector is your eye or whatever is a photo, whatever is being used. Here is what it looks like if these 1 micron spheres would be emitting fluorescence. You can see the difference and how you can identify the different colors. You can quantitate those. The light is passing through the sample, it's diffracted and absorbed, and then it's filtered to detect light from different molecules within the system. This is a great example of how you can detect differences even at the 1 micron level just by using fluorescence, not adding anything else or doing anything different than adding fluorescence to the sample. All right, so what is fluorescence actually used for? Great question. First, it is used for nanotechnology imaging. Fluorescence is a technique that is widely applied in imaging and diagnosis. The applications include a study of enzyme activity, identification of tumor and cancer cells, and other disorders. This technique can also use to apply to study the phenotypic changes in intercellular organelles like mitochondria and nucleus. What is actually happening and what is changing in those organelles at the cellular level. Now, the photo on the right here, you can see that this column here is a transmission column, and this column here is the fluorescent column, and then this column is a 3D column using the information from transmission technology, fluorescence technology, and adding it together to find what the 3D image actually looks like. Now, these are samples of blood cells that look differently. You can see that they look differently in the transmission and the fluorescent, and then they have used that information to make it into a 3D model. Now, not all of these look that different, but especially when you get down here and you can see that there is something inside this blood cell that is fluorescing differently than what is difficult to see in the blood cell on just the transmission level. And here also, you can see this on the transmission level, but it shows up so much better on the fluorescence to be able to see what is actually happening in those cells. All right, the second technique that is used in fluorescence is amazing and is typically called FISH. That stands for fluorescent space in situ hybridization, aka FISH. And it's another technique that is very reliable, precise, and sensitive. It is typically used for DNA testing. It's used for trisomy 21 testing and prenatal testing when they take amniotic fluid from the mother and run it to see. It's also used to study abnormalities in related to chromosomes 13, 18, 21, X, and Y. The reason these chromosomes are studied the most often is it seems that this is the most often that there may be abnormalities that the fetus can survive and would be born with. So if you look at the photo, you can see that the left-hand side are the chromosomes under fluorescence. The right-hand side is the photo of chromosomes using the electron microscope, and you can see the bands on the chromosomes on this side. And then the arrows are pointing to areas where there possibly is abnormalities. Obviously, on this side, they're hard to see. Like, it would be very difficult for me to pick these out. But on the fluorescent side, those abnormalities are like flags sticking out and being shown. Right here you can see where those abnormalities are found, and make it easy to see and diagnose for doctors and to have less, problems or issues missing something. All right, the third thing that fluorescence can be used for is looking and being able to see the timing of protein folding and unfolding. How does that work? Well, protein folding and unfolding is sensitive to environmental changes. We know that. Fluorescence is easy to use. Typically, they use some type of dye. In this example, they're using the Spiro orange dye. And you can see the protein and ligand interaction, both quantitatively and qualitatively. The x-axis here is the temperature, and the fluorescence is here on the y-axis. You can see that at a lower temperature, the protein is folded nicely. There is no dye attaching to it. But as the temperature begins to rise, the protein begins to unfold, and as it begins to unfold, that dye begins to bind. As the dye begins to bind and fluoresce, the fluorescence increases, and you can see that it peaks up here at the top where all kinds of dyes are binding to that unfolding protein. Then the protein begins to completely unfold. Less of the dye is bound to the protein as it's going down, and then here the protein is completely denatured and there's very little dye bound to it. But you can actually make a graph and be able to see the fluorescence and how that is happening. There are limitations to fluorescence. First of all, not everything can fluoresce. It's very difficult to measure something if it is not fluorescing. Secondly, there is photobleaching, and this is a process when the dye that is used becomes non-fluorescent due to something in its molecular structure. It gets altered as a result of the exposure to light and can sometimes give false results. Another thing is phototoxicity, and that is the process when the cells become damaged due to the interaction between the fluorescent dye and the excitation of light. Phototoxicity can give false results. It can damage the sample, all kinds of things. Sometimes the amount of light that is emitted is low, and that can lead to a dim or blurry image that's difficult to see. It's hard to use a dim or blurry image. Also, some fluorochromes emit light at different wavelengths under different situations. So it's really, really important that the correct spectrum is used, because it could give one light at one wavelength and it could give a different light at a different wavelength, which could lead to false results or a misunderstanding of what is actually being seen. All right, so the references that I used were fluorescence quenching theory and applications, the principles of fluorescence spectroscopy, the measuring structural changes in proteins, genetically encoded fluorescent tags, molecular biology of the cell, and the fluorescent optical tomography for cancer detection. Thank you so much for watching my presentation. Have a great day.